Cell signaling pathways. Cell signaling steps. Cell signaling consists of 4 steps. 1. Signal travels to receptor. 2. Signal alters receptor. 3. Signal is transduced. In this case, transduced means to convert a signal into another form. So it is transduced through the cell. And then 4, the cell responds. Other examples of cellular communication which do not include signals, such as delta and notch, are beyond the scope of this course. Location of receptors. Ligands, which are polar, hence water-soluble, cannot pass through the cell membrane. That's because they're blocked by the hydrophobic portion of the membrane, indicated by the black arrow. Non-polar ligands, for instance nitrous oxide, can pass through the membrane. So, receptors for nonpolar ligands, like nitrous oxide, are found within the cytoplasm of the cell. Receptors for polar ligands have a binding site outside the cell. The receptor passes through the cell membrane into the cytoplasm, making it a transmembrane receptor. When the ligand binds to it, the portion of the receptor inside the cell changes shape, communicating the message into the cell without matter passing into the cell. Transmembrane receptors are composed of proteins or glycoproteins that are anchored in and extend from the cell membrane. The process of a signal being carried into a cell by a transmembrane receptor is called transduction. Transduction signaling. In this example, a polar ligand, indicated by the red triangle labeled 1, travels to a cell where it binds to a specific transmembrane receptor, indicated by the blue Y labeled 2. What that does is changes the receptor's shape. A signal is transduced across the cell membrane and the cytoplasm with the help of second messengers, shown by these yellow pentagons. The cell responds by changing its cytoplasm enzyme activity and or its gene expression pattern, indicated by 4. Second messengers. Second messengers, which we've said before were indicated by these yellow pentagons, are small molecules or ions that continue signal transduction pathways in the cytoplasm. So remember, the receptor changes shape, and that passes the signal to these second messengers, which can then continue the pathway in the cytoplasm. Some second messengers can interact with many molecules, amplifying a response, initiating multiple responses, and interacting with other signaling pathways. One very important second messenger is CAMP, cyclic adenosine monophosphate. It is related to these molecules: AMP, adenosine monophosphate, ADP, adenosine diphosphate, and ATP, adenosine triphosphate. CAMP can be produced by reactions involving AMP, ADP, or ATP. Signal is transduced. Signal transduction pathways convert the signal into a cellular response. This begins with a change in the receptor, which then results in changes within the cell. While there are thousands of signals and receptors, there are many fewer transduction pathways that carry their signals. In some cases, a single receptor activates multiple pathways. In other cases, multiple receptors work together to activate the same pathway. How the pathways and receptors are interconnected defines how the cell responds to signals. Cell responds. Cellular responses to a particular set of signals could potentially include everything cells do, such as activation of cytoplasmic enzymes, change in gene expression, entry into the cell cycle, pause in the cell cycle to repair DNA or organelles, apoptosis or programmed cell death, maturation or specialization of cells, movement, migration, or shape changes of cells. Internal signaling. Critical signals also come from the cell's internal environment and don't involve cell surface receptors. For example, damaged chromosomes lead to signals that cause cells to pause and repair. Internal signals and responses are crucial for the maintenance of homeostasis. Environmental signaling. Signals can also come from an organism's external environment and received by its cells. Pheromones enter the organism, diffuse across the cell membrane, and bind to internal receptors. Heat shock, 108 degrees Fahrenheit or above for human cells, causes changes in protein shape and folding that signals other responses. Electromagnetic waves can be signals. For example, light waves interact with photon-sensitive molecules inside the cell, such as photoreceptors or pigments, and initiate a response.